So again, the reason why we need a minimal length of a membrane helix is because you simply have to span the roughly 3 nanometer membrane. There are slightly different membranes. Autobacterial walls have thicker membranes, but most membranes in eukaryote are about 3 nanometer, 2.5 to 3.5 nanometer. And that is essentially from 20 to 25. And that is essentially exactly what you see here. So the peak of the distribution. Now, let me sort of illustrate a little bit what the meaning is here. You are late. I thought you were under the tripod. So, red says low here. What low means, essentially, that's low resolution experiments. Do you have any idea how low resolution experiments measure where membrane helices are? Essentially, they label something outside and then they have kinase digestion and you see some light, so whether it's outside or not, and this way you sort of figure out roughly where the membrane helix is. And they're low resolution in the sense that this is not a crystal structure. Blue is the crystal structure. And that is data from 14 years ago. But you see that the blue, the red is what we expect. This is the whole story I told you. It's the story that makes a lot of sense. Blue is very, very different from it. So this one is showing the same sort of distribution, but now cumulative. And in the cumulative distribution, you see that, so I said they are between 20 and 25. In blue, you see that half of all the membrane helices are longer than 25. So 20, in this distribution here, 20 to 25 captures roughly 80% of the distribution. So essentially, this is the dominant thing, plus minus 1 on either end, and you essentially have the entire distribution. For the blue, that's not true at all. Now, it is true that in the crystal structure, we don't see the lipids. So the placement of the membrane helixes there is not completely decided. But this still is extreme. So how could we imagine that we have a very, very long membrane helix? And here's an example like that. In this example, do you see the membrane? So, I mean, it's not drawn there, but do you imagine that you see it? Have you seen enough of these pictures to jump up and say, okay, my lipid bilayer is somewhere here and here, and in between is the lipid bilayer. Equally here, we somehow see it here and here. And the helix under investigation is this one here. It's way, way, way longer than it needs to span the membrane helix. And actually you see it sort of extends into the globular region of that protein. Which means without any interruption here, you just have a very, very, very long membrane helix. Or this is not a membrane helix anymore, this is a globular helix here. But it's just the same way. You have something that's on the one side in the lipid, on the other side inside of the protein. In both cases, you want a hydrophobic environment. So for that helix, the environment is not that different. One is in terms of function, or in terms of structure, in terms of the way we interpret hydrophobicity here, is very different. This is a membrane helix, this is a globular helix, right? And that is very, very difficult to distinguish. In the first type of structures people did, they were more of this type. They were simpler. Later we saw examples like this. And Gunnar van Heijnen has recently seen one example where, in fact, there was something that was completely outside of the membrane region that looked like a perfect membrane helix. And for many years he looked at all kinds of prediction methods, and for many years he didn't understand why. And then he found one confirmation in which that helix actually was in the membrane. So in a situation in which, so this is not the case that I show here, in that case that I'm talking about, there was a membrane helix that sometimes was not. Okay? So membrane helices are also obviously things that, maybe because of a similar reason here, maybe because they can hide inside of proteins, or hide is the wrong word here for hiding, but they are also happy inside of a protein, and that is why there are sort of two different states. And maybe we should go into the world where we essentially distinguish between different types of helices, membrane helices. Those that are always in the membrane, and those that sort of can be more mobile. Unfortunately, the data is not enough for that. Now, what this slide shows you is that, and let's not get into the detail, that essentially is the difference in performance. And essentially what it says is that the longer the membrane helix, the more membrane prediction methods make mistakes. And that's clear, because they have learned that membrane helices have to have a minimal length, and if I make them too long, then I have to cut it. That's what all the methods, the prediction methods, focus on. So here's another issue. When you have a membrane helix, another membrane helix, and you have a certain connection. So now I'm asking, what is the how long is the loop in between? And again, for the red one, you see that most loops that connect membrane helices are sort of 5 to 15 or something like this. So they're fairly extended. So you have enough sequence signal to say, okay, this is hydrophobic, there's something else, that is the loop that connects, and then we have the membrane, the hydrophobic helix again, right? It's a very clear sequence signal. But when you look at the sort of more modern structures, you see that the connection is shorter. And in some cases, really very, very short. How can a loop be one residue long? Can it? So when you observe something like this, so here's an actual data point, and the data point is actually one residue, right? And this is a certain percentage, it's not zero. So what's your conclusion from something like this? It cannot be. In fact, even I would argue two cannot be, but because you have to have something that really spans between two different helices that orient in different ways, up, down, and you have to connect it, right? So with two residues, you still hardly can do that. Okay, to proline.
Couldn't we just abandon the helix like this?
That could be one way. So you have essentially, you don't have a straight helix, you have a sort of crutched, crooked helix. That is one way. But when you see data like that, the first thing you would actually do is check out your program, that this is really right. In this particular case, it is really right. And in fact, in this particular case, we were not crooked helices. What could it be?
The crystallographers had to cut out the loops because they had a too bad resolution.
This is, in fact, some of these were exactly that. But we also saw some where our interpretation, at least, was that the helix moves. Okay, so that the hydrophobic cuff that I showed you before is an approximation. And now when you talk about one residue loop, whether you're 1 or 2 off is a big difference. Suddenly you move this to a 3, and then, well, we form a loop that is possible, right? So it's not quite a mistake. It's not quite a crooked helix, but it's actually something that has to do with essentially the flexibility of the way membrane helix is inserted. But again, what we see on this slide here is that for the ones our prediction quality increases, so the prediction accuracy is much higher for longer loops. Okay? For short loops, we make mistakes. So those are features of membrane proteins that we had not really anticipated, and that make it more complicated. This is another issue that has to do with the evolutionary conservation. So say, do you know the answer to that question for human? How many membrane proteins are there?
A quarter.
A quarter. And you know that because a quarter is a good number, or you know that because somebody has told you?
I think I heard somewhere.
I'm just curious, where would you hear something like that?
Like
If somebody gives a talk?
a couple of lectures ago.
For me?
Yeah.
Oh. Oh, you have listened to me? No, that's bad. Have I said that already? Okay. Ballpark 25%, yes. But how would you actually go about answering that question? I give you a method that predicts membrane helices. Did we talk about that? So you have a method. One way is the one that Jonas or Michael presented to you last Thursday. complex method, much simpler neural network method, whatever you want. You have a method that predicts for every single residue in the protein where the membrane helices are. Now you want to apply that method to establish how many membrane proteins are in human. How would you do that? So let's begin with the simplest thing. Where do you get your sequences for human from? From a database. Can somebody name that database? Okay, I'd argue this is simple, or Hauke, you're always the one who tells me it's simple or not.
I would have said Uniprot.
So that means it was complicated now? Or because you got it right, must have been complicated? I don't want, I'm sorry, I need interaction, but I don't want to put you on the spot.
I think sometimes we just sit here and think, is it Uniprot? No, it can't be Uniprot, it's too easy. And then nobody says anything. That's what happens.
Yes, because most of the time I say no. It's true, most of the times I set you up. Okay, it's my mistake then. So you begin with 20,000 proteins in Uniprot, or Swissprot in fact, in this particular case, and then you predict for every single one the membrane helices. Is that enough? No, because now actually, so what we have developed so far are methods that are optimized to finding the membrane helix in a membrane protein. They are not necessarily the best to differentiate between a protein that has membrane helices and not. Okay? This is a subtle difference, and it may well be that the same method, in principle, if your method worked very well, you would expect that the same is right. But some of you in the last semester, remember, we talked about protein interaction prediction. And for protein interaction prediction, to identify where two proteins interact and which two proteins interact, those are very different methods. That doesn't make me happy. I cannot explain why that is the case, but it's an observation, and it's similar, it's sort of similar thing here. It's slightly different. So the simplest way of doing that is, so if that is the method, were the method that predicts membrane helices, and you have some threshold here, that could be 50% or could be somehow higher, and the threshold would be such that you would say this is 1, 2, 3, 4, 5. Membrane helices. But now the question really is, if my protein had one membrane helix, all you need to go for the thing that looks like a membrane helix with the highest score. At least one or not. That's the distinction between membrane protein and not. Right? So you essentially look for the highest area under the curve here. And by the way, look quickly. Sorry for the animation. By the way, when we look at number of residues in proteins, percentage of proteins for different types of proteins, or kingdoms, archaea, prokaryotes in green, and eukaryotes in blue, essentially, proteins are similar in terms of the length distribution. You knew all of that, right? Good. So some people say that eukaryotes are longer. Essentially, it's not really true. The peak of the distribution. So here, by the way, when you look at these distributions, you immediately see that quoting a protein to be 230 residues long as an average or something really doesn't make sense. They are between 100 and 400. There's a big average in there, and any way of compiling the average of a protein length is really not meaningful. There's just a big distribution in some region between 100 and 400. And in fact, that is really similar between the kingdoms. What is different, however, is when you look, and that is cumulative now, at the ones that are longer than, say, 1,000 residues. For archaea and prokaryotes, this is about 1%. For eukaryotes, this is about 7%. So where they do differ is not here. Where they do differ is they have many more of the very long ones. Okay, that is specific, and again, the longest protein known as titin in human. When we look at the overall amino acid composition, height of the letter proportional to the usage of that amino acid for the three different kingdoms is similar. Now this is somehow surprising, isn't it? Because you do know about the GC bias of different organisms. So different organisms really use different codons. But on the level of the amino acid, this is hardly visible. Again, there's a signal in the detail. You can use that signal. That's a paper that was published by Joel Sussman's group at some point. But nevertheless, it's in some sense, I would argue it's surprising that overall on the level of amino acids, there's no clear signal. And there's also not really a signal on the level of family sizes. But let's get back to the membrane issue. Here we have sort of the 25% that you talked about. And for different organisms, you see that essentially they have, do they have similar ones or not? So that's the composition of membrane proteins. Proteins with helical transmembrane, with transmembrane helices in the entire organism. Eukaryotes, prokaryotes, archaea. Are they similar? Are they different? Whatever you decide is right. Just argue for it. So obviously nobody can in their head, but now maybe you have had the time to actually compute these numbers and add them up and do it. Maybe some of you are polymaths who can do that sort of thing. Nobody can do that. But my point is, I would argue they do not really differ, because the variant within the variation within one of these proteomes is so high that the difference in the average is much less significant in some sense. So we clearly do not see a difference in sort of the membrane composition. And you may argue this is understandable, because after all, everybody has sort of the same reason to let some stuff get in and some stuff get out, and it's a similar fraction. But you may also argue that, in fact, initially people assumed that human would have a higher ratio, because that has to do, in some sense, with a higher complexity of the cells, a higher level of communication between the cells. Clearly, eukaryotes have a higher level of communication between cells than prokaryotes. But ultimately, this is not visible, at least on the level of what fraction is transmembrane helix. Another thing I can look at is the number of transmembrane helices in a protein. And the first, so that's 100% of all my proteins, and now 100% of all the membrane proteins. And I do have a problem here somehow with a graph. Maybe this is everybody has at least one. Maybe that's where the numbers map. Is that visually for you okay? Because where I'm standing, it looks like 2 up there. Yeah, it is 1. Okay, it's just my position. So 100% have in this sense, in this set here, have at least one membrane helix. Now, what you see is 2 is sort of roughly 40% of them is gone. So most membrane proteins, or proteins with transmembrane helices, have 1. Not most, it's less than 50% here. But the class with the highest, so the single highest one is the ones with 1. That's what I'm trying to say. The other simple observation again is eukaryote prokaryotes are similar. There's no difference. And then, you just have sort of an expected fall off. There are proteins with more here, but they just don't count in terms of the overall numbers. Now, however, and now we get into the issue where you have to just believe me. Now we get into the world where we essentially look at 2 different orientations. Is the N-terminus inside or outside? Does my protein start the outside of the membrane or the inside? In is white, out is black. What we see is the composition for proteins with 1, 2, 3, blah, 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 membrane proteins, membrane helices. And you see that here's a peak. A 7 out, and that's the GPCRs. You see a sort of similar peak. Well, no, you don't actually see it anywhere else. You see peaks in bacteria that are at 6 and 12. So here is actually Drosophila, and Drosophila has the GPCR. C. elegans doesn't. But you see there are certain, wait a minute, this is Drosophila. This is C. elegans, and C. elegans has this scent. Yeast doesn't. But you see immediately some things stand out. Again, I understand that you don't see. Believe me, it's an old slide. But on a level where you remain as simple as just look at the topology and the number of helices, you see some functionalities that have to do with the level of the organism. Most, it's too complicated. And I believe, coils. Well, coils are historically named, Francis Crick invented that name, and essentially, I would call it a misnomer. Essentially, you have two long helices that are intertwined. The leucine zipper is the example where you use it to bind DNA. Julian Foss is another example to bind DNA. Here, keratin filaments, very, very, very long fibers. Here we have an example where essentially you have a membrane helix anchored. You follow it by a coil. Here, membrane helix followed by a coil. Now you wind these two coils. So what do you believe is happening? So you just twist these coils. What is going to happen?
It's going to return back, right? It's going to return back physically, right?
So something, there will be some counter reaction, that is true, but let's just assume, so, okay, these are my two cell walls of my cells, and now I have my coil, and now I intertwine them. What is going to happen?
Stay together.
They stay together, yeah, but what is going to happen to my cells? Voilà, did you? No? They march, not exactly march, but yes. So they approach. This is exactly the way in which you bring cells together. So you essentially twist a coil. I mean, this is very visual in that sense. You twist coils and you shrink the space. And you can, now in this drawing you don't see that, in this sketch you don't see that. But you could imagine that they're very far apart because these coils are very, very long. And then they begin to pull them by intertwining. Okay? At some point you're right. At some point it will go away again. But this is essentially the way you make essentially cells approach each other, sort of dock. Coil, again, overall for organisms, there are more coils in eukaryotes, but not much. They're longer. That's the composition of extracellular proteins, that's the composition of nuclear proteins. Let's get into the issue, the question of how many membrane proteins do we know structures of. Today, we believe that about 20-something or 25% of all human proteins are membrane proteins, but for fewer than 2% of those, we know the structures. And that is essentially, as I told you, because we, in fact, it was so difficult to do the structures. And this is just one slide from a consortium. Edda and Marco in our group worked on trying to identify from all the families in human what are the most promising candidates for, that experimental collaborators could possibly get structures for. There was structural genomics for membrane proteins, and this consortium in New York determined, in fact, a relatively large number of membrane structures. So the large number is 20, but and that was done over the course of 10 years. But in the membrane field, 20 was a big number. And made a remarkable distribution. The consortium was led by Wayne Hendrickson. And I'm going to leave it here for membrane helical proteins.